My name is Andy Breakspear, and I work at John Innes Centre, and I'd just like to thank Devin for inviting me here today. I have to talk a little bit about the biology of the beast that we started to work with. This is Medicago truncatula, but importantly, it's a legume. It's like peas and beans, and it's got one very cool trick up its sleeve. So actually, it's able to team up with a nitrogen-fixing bacterium, Cinorhizobium meliloti. And the plant actually feeds this bug sugars, and it quite happily does that because in return, the bacterium provides a usable source of nitrogen to the plant. Okay, and this all goes on in a nodule structure which are found on the roots of the plant here. So this is a real bonus for this plant actually, because nitrogen is a very unreliable nutrient in the soil. It's here one moment and it's gone the next. So for a legume, the ability to team up with this bacterium is a great thing. But unfortunately, it's restricted mainly to legumes. So cereal crops like rice, wheat, maize, etc., they can't do this. So to grow a cereal, if you want to get the best possible yield, then you need to apply fertilizer to grow your cereal crops. Fertilizer is environmentally damaging, and to many smallholder African farmers, it's simply unaffordable. So I just want to quickly show you the pathway that the bacterium takes to get into the plant, because it's quite remarkable, and you'd never imagine this. So first of all, the plant and the microbe kind of taste each other. They secrete some signals and they recognize, and if they like each other, then the plant will initiate a program to accommodate that microbe. And it starts off with, so this image here is of a root hair. This is the root here, the main root. The blue staining are actually bacteria. And so it starts with the bacteria binding these root hairs. Okay, they stick on tightly. And then the plant root hair starts to curl around the bacteria and it grabs those bacteria tightly. And that's what you can see here. This is an infection focus, or what we call infection focus. Sometime shortly after that, the plant starts to build this tunnel, or infection thread as we call it, that burrows down the length of the root hair into the middle of the root. And in the middle of the root is where we get the initiation of these nodule structures, where we get they're packed full of these bacteria, and they're fixing nitrogen into a usable form of nitrogen that the plant can use to start building its proteins. So this is the pathway that it takes. But as I said earlier, this is mainly restricted to legumes. It is not in cereal crops. So it's been documented for quite some time. Can we put the ability that legumes have, peas and beans, to team up with the bacteria, can we transfer the genes required to do this into a cereal crop so we don't need to use fertilizer to grow a cereal? So it's been known for a long time that this is obviously a good, an exciting way forward, but the technology wasn't really there. We're talking about a project that needs the introduction of maybe 50 genes to get a cereal crop that might be able to fix nitrogen. So we felt about 4 years ago that the time was right to give this a shot. And so the Engineering Nitrogen Symbiosis for Africa project was set up with a grant from the Bill and Melinda Gates Foundation. It was a super exciting project, really motivating for me to come in and work on this. But it was a wee bit daunting. Many people have transferred single genes or even a couple of genes into plants. So we were looking at, transferring many dozens of genes. And so we didn't want to do this in isolation. And so we teamed up with two other large engineering projects at the time, and they're still running. This is the C4 Rice project and the Wright project. And both of these are either engineering a new pathway in a plant or heavily modifying an existing pathway. And so we set up, about 3 years ago now, we set up the Crop Engineering Consortium. And the goals of this actually were to let's get together and let's share our results, let's share our data. We don't want to be making the same mistake twice. And if we get good results, we don't want to reinvent the wheel. Let's share the technology that we're using, and that's successful. And so, you know, I think it's been so powerful to get together and have this kind of attitude, and I think it really allows us to advance more quickly. So we settled on an engineering strategy towards the beginning of the project, and it starts, like most people's, with synthesizing these DNA parts we heard a lot about yesterday. You then put these parts together in a single pot reaction, to wrong button, okay, to produce your construct that you want to transform into the plant. We always resequence everything we do because of the costs associated with transforming a cereal crop. I don't want to be responsible for a mutation, you know, 2 or 3 years down the line. Andy, didn't you design this one? You know, for a little bit of organization now, it's well worth that. So transformation, but then the exciting bit that most scientists enjoy is actually looking at those transformed plants. Are the plants doing what we hoped they would? If they are, then that's great. If not, we need to look at the design and think about what we're going to do next. So that was the general cycle that we thought we'd agree on. And in just ENSA alone, in the last 4 years, we've produced over 2,000 transgenic lines in barley. So barley's our model organism. We've got a really good transformation pipeline running at the John Innes. And this is really the result of, it's a lot of hard work from the individuals, but it's been great to be part of that consortium, sharing DNA parts, sharing experiences. But we did feel that actually we're not doing things as efficient as we could be. And that's when, about a year ago actually, we sat down with people from the Earlham Institute and we thought, well, you know what, actually most of the scientists, they really want to be focused here and here. And it's kind of fun to do this Golden Gate assembly a few times, but after you've done that, you know, it gets a bit boring. So we wanted to free up an RA or a postdoc's time from this part of the circle, the cycle, and allow them to focus on this bit, you know, the really exciting science. This also had the bonus, of course, of doing assembly much more efficiently, much more quickly, more cheaply. So I'm just going to take you through this synthesis, assembly, and verification pipeline. And synthesis actually was being done very efficiently and very well by a company. So we've pretty much teamed up with Life Technologies. And you know, they're more of a partner than anything else, I have to say. So we've got a very good rate on synthesis because we have a consortium and a lot of products that we want to synthesize. And then we have a lot of bargaining power, a lot of purchasing power. And so we were able to reduce those synthesis costs. And what that did was it allowed people that weren't synthesizing, that couldn't really afford it, to afford it. And it allowed the people that could afford some synthesis to do a lot more synthesis. So actually, it turns out it's cheaper to get stuff synthesized than it is to be cloning in the lab, to be doing the PCR and the resequencing. It's better off that you focus your time elsewhere. So we've done quite a bit of synthesis, and we're really pleased with letting the company take that part. But what's been great is that we've got a centralized resource now. All those parts that we get in are all put in a single collection at the JIC that will shortly be transferring over to the Earlham Institute. And this is great because it keeps us all on the same page. All the DNAs are of the same concentration. They're all well described, etc. Whereas if you give bits out and everyone manages their own, then you have this inconsistency. And when it comes to swapping, you can have some problems. So what we've got now is this really amazing collection of parts. And this is just a snapshot of the parts that we have. So we've got around 5,000 of these small DNA parts. And these can be promoters, coding sequences, tags, epitope tags, fluorescent tags, etc., and a bunch of terminators as well. So as people have explained yesterday, so we're using the Golden Gate platform, and you can very easily pick any promoter here and any CDS you want and join them with any tag or terminator in a single pot reaction. So it's super flexible. But the other great thing, I think, and the thing that I really like, if you, for example, you want a promoter for constitutive expression in maize, you can go and have a look and you can see, oh, this one's being used. It said, oh, the previous user said it works great in barley and rice, but it's terrible in maize. If you want something that works in maize, try this one. It's great. That sort of information is invaluable when you've got a platform where the transformation, can take, up to 9 months to generate your transgenic crops. You've then got to wait another generation to take them through to homozygosity. So the information that we're building up, of each individual parts of these collections, I think is a wonderful thing. Of course, any good collection of parts needs a database to look those parts up. So we have online database, and every DNA part that we get synthesized gets numbered and added to the database. On the database, we have a complete plasmid map. Everything is resequenced, so we're happy with the sequence that we have. Gone are the days of plasmid maps on the back of an envelope with a few restriction sites to get you started. It's all there for you. And then, as I said earlier, for every plasmid that's being transferred into a plant, it has a link to those plants, and you can see how that particular plasmid has performed in that plant. So, I mean, we are in the early stages, but we're starting, to build up, get an idea of what works well and what doesn't. So in the simplest case, then, you want to pick a promoter and a CDS and a terminator. You can go ahead and very easily do that from the collection. Add that to a backbone vector, and you put it through the Golden Gate pipeline. This is well known. It's very quick. It's a 4-hour reaction, and you'll get a single gene construct produced here. You can then take that single gene and you can combine it with up to 5 other genes that you've either just produced or they may be already in the library. And just as you can with the basic system, you could put them in different, or any orientation you want. You can put them in any order you want on the construct. And then you run them through a very similar pipeline to the previous reaction, an almost identical pipeline, and you can make a multi-gene construct that then you can put straight into the plant. So the technology that underpins this is actually really basic, and it consists of a freezer to store your modules, and you have a pipette and a thermocycler to carry out the reactions. And then there's a bit of basic microbiology to propagate the plasmid and then extract the plasmid. So this was great in the very early stages. As a technician such as myself could put together maybe around a dozen of these per week, maximum. And it's a little bit risky because just to do a dozen constructs, you're looking at, and it does depend on what you're doing, but you could be easily looking at more than 500 single-channel pipette deliveries. This, what we do, these are bespoke reactions. Each reaction has different promoters, different CDSs, tags, etc. So they don't lend themselves well to multi-channel pipettes. But of course, projects now here that require, including ours, that require hundreds, And now people are talking about thousands of constructs. So clearly we needed to automate this process. So we sat down with the folks at Earlham Institute and looked about how best to do this. And this was, as I say, about a year ago. And we split it into modules. And it was really centered around that ability to take those catalog library parts and mix them in a bespoke manner into a destination plate. And so the Echo liquid handler, is really the center of our pipeline. So we went for the access system, and we have integrated a plate sealer and a centrifuge and a thermocycler, and so we can essentially feed the Echo our catalog of parts, and then have out our ligated products. So just to give you a better idea of that, so we'll have our library modules contained within Echo source plates. We use 384 low dead volume plates. I think these are 1536. So we feed them into the Echo, and then what we'd get in each destination well, we'd get a mixture of those parts, which were then, the Diglig reaction then happens on the thermocycler, and then we get out the reactions ready to transform E. coli. So as many have alluded to during this symposium, one of the great things here is the ability to miniaturize the reaction. And so when we were doing this manually, we'd be using 15 microliters. And I was chatting to the automation specialist at Earlham Institute, Tony West, just last week. And he was now looking at 500 nanoliter reactions for this. And his initial results look really promising. So, I mean, we're really saving a lot of money on miniaturizing here. So we're really excited about that. This is just a brief description of the very first project that we ran using our robots. So I said before, we're taking genes that occur in a legume and that we know are important for forming the symbiosis with the bacteria. And we're transferring those genes into a cereal crop, into barley in the first instance. But in order to get correct expression of these genes, we need the correct barley promoter to do that. And so we found it very difficult to find suitable promoters. And so really it's a case of trial and error, to tailor the expression of these transcription factors correctly. And so this was our first experiment. It just takes 20 different promoters, and it combines each one of these promoters with a transcription factor, CDS for a transcription factor, and we fixed in the terminator to make life a bit easier in this first instance. So we're looking at a total of 180 constructs because as well as just doing a single gene of interest, we wanted to test 6 genes of interest in one go here. So the layout on an Echo 384 plate will look, well, it looks just like this. With these gold boxes are for some backbone components. The green boxes are those promoters, those 20 promoters that we're testing. The blue boxes are the CDSs, the genes of interest, the transcription factors. The orange boxes are terminator, and a few other bits and bobs required for the reaction. So we're asking, in this project, we're asking the Echo to do 720 deliveries here. So if you're familiar with the software, this is using cherry pick and would import a pick list of 720 lines. And in this particular case, this involved 57,600 2.5 nanoliter shots. We didn't miniaturize as much as we could in the first instance. This took 8 minutes, so 22 constructs a minute are being mixed. So, I mean, this really, really changed the way we could work. And, obviously, if I was preparing these, then, maybe 1 every 22 is going to be incorrect. I'll tell you now, from doing the sequence verification, 100% of the 180 were correct. So here's what we'd get out of the Echo, the destination plate containing bespoke mixtures in each of these destination wells. So we wanted to really support the echo at either side. So first of all, our domestic freezer, you know, is great on a small scale, but if you can imagine digging out 200 parts out of a freezer and then putting them back in the right order, it starts to take quite a bit of time. And so we are about to receive this automated freezer with cherry picking capability. So you can dial in what you want the day before, and then overnight the freezer will sort those modules out for you. You come in the next morning and it delivers your modules in as many racks as it takes. This particular freezer has room for 70,000 0.7 ml barcoded tubes. And what we do or what we will do is team this up with a traditional liquid handler. This is from Hamilton. And we use this now without the freezer. But what this module is really designed to do is to decap the tubes in an automated fashion. The decapped tubes then get shuffled out onto the deck, and the robot then creates the source plate or plates for the echo. It's brilliant. Before we had this running, I was preparing the echo source plates manually. It takes a lot of concentration. If you're doing that, you know, get your noise-cancelling headphones. If you're lucky enough to have a robot to prepare it for you, then you know it really is super. And then what we were getting out of the echo, those mixtures, what we wanted to do was to actually try and automate some of the microbiology that we needed to do. And again, we can use the Hamilton deck for this. So this is currently a work in progress. Tony West at the Earlham Institute is working very hard to finish the programs for E. coli transformation, colony picking, and a 96-well plasmid prep. So we're getting very close now to running and using this functionality. We're working in 8-well plates, as you can see here, which works very nicely. So that's our pipeline. So yeah, we're looking at really hundreds of constructs per week if we want. We want to, you know, we're super lucky at the Earlham Institute because once we've generated these constructs, for sequence validation, it's simply a case of knocking on the glass and waving to Darren on the other side, you know, with his Illumina machines. And he runs our samples in batches of 96. And as I said, in that first project that we did out of the 180, they all came back as we'd expected. Since then, we have had some failures. And I'll tell you where they come from, and it's all from one particular reason, is if you use multiple copies of the same DNA part, then you're inviting recombination in bacteria. And that's what we found, that we were getting looping out of large parts of the construct. So I'd thoroughly recommend, you know, not repeat. If you're using the Golden Gate system, don't repeat these parts. Now, I was worried about it because I thought you might get silencing in the plant, but actually the problem comes earlier than that, and it's a recombination in the bacteria. So that we needed a bit more of a traditional screening of the white colonies for those, but we got through it very quickly. The consortium that we originally had 3 years ago has expanded now to include members from these institutes and organizations. And I wanted to highlight one in particular, and that's Biosciences Eastern and Central Africa, or BECA. So these guys are based in Kenya, in Nairobi. And what I found is, and it should be fairly obvious, that actually there's no one more motivated to tackle starvation in Africa than Africans themselves. And so going over to BECA to see that motivation, you know, it was a wonderful thing. And, you know, taking over and sharing some of the technology that we'd developed with Golden Gate, it was like, you know, providing them with this kit to very easily put their dream constructs together. And so we've really got a lot out of our relationship with BECA. Of course, it doesn't really matter how good your platform or pipeline is. you only get good stuff out if you put correct designs in. And so really an integral part of what we've been doing is to provide training to break the access barrier and to understand how Golden Gate works and how you design a construct. So we put together an instruction manual which covers the whole process of designing these things in silico to actually building them in a lab-based environment. So we do a 3-day course, and we've done this twice in Nairobi. So Becker is a capacity building hub, so institutes from other Central and Eastern African institutes come to Becker, and they can learn a technology that they can then take back to their partner institutes. And so by going to Becker, we had the ability to reach out to many institutions. So we learned a lot from this, and I think the major criticism of this instruction booklet was that we were using commercial software to design the in silico maps for our constructs, which is a great way of checking your designs for Golden Gate. And so we replaced those bits of software with freely available software, so you could build, so anyone could really, that had access to the internet could build these constructs. It's not a completely selfless thing, training other people to do this. It is, a lot of fun, but we get back a lot of basic parts for the library, and, with their permission, then they can be shared with other members of the consortium. But perhaps more excitingly, you'll have someone will say, Andy, this is great, but actually I want to do CRISPR with the Golden Gate system. How do I do this for CRISPR? What modules do you have for CRISPR? And I'm like, oh, we don't have any CRISPR modules. So we sit down and we work out a strategy of producing constructs for CRISPR using the Golden Gate platform. And so this has expanded. I know there's someone here that loves RNAi. Well, you're not the only one. RNAi is very much in demand from us. And so we set up a number of constructs to help with RNAi designs. Others on there, we had the viral 2A system, so you can drive many eukaryotic genes with a single promoter. So just to finish, I wanted just to summarize the platform. So I really am, this is a hobby for me, building constructs. My day job is doing RNA-seq experiments across the road. But every now and then I pop down for some therapy and come and use the robot. So we've put together 840 sequence-validated constructs since getting the first robot in April. With just me operating this, we're looking at 15-day turnaround, and most of that is coming from sequencing the intermediates that we're producing. We've got 2 future projects that are being put into fluid X tubes. Right now, that's one is a CRISPR, large-scale CRISPR project, and another is a domain shuffling experiment, so making a gene and mixing around these domains to try and optimize that gene. So that just leaves me to thank, first of all, my supervisor, Christian Rogers, and Giles Oldroyd, who won the Bill and Melinda Gates grant for the ENSA project. I've been really lucky to walk into this. It's a fantastic project. I really feel motivated by it. I'd like to thank, Doreen, Eleni, and Andre. These are other RAs that I work with on a daily basis. And the folks over at the Earlham Institute that I work closely with, Tony West, who's the automation specialist. And it's brilliant because I can go and use these. If I get stuck, I can put my hand up. Tony's really responsive, comes and helps quite often. Dan Swan. So I'd like to thank Dan for those initial meetings of setting this pipeline up, and quite frankly, for letting me come in and using your robots. Kirsten, for managing the platform and helping get set up, and Darren Heavens for doing all of our resequencing of our constructs. And then finally, I'd like to thank Devin for convincing us that an echo was what we needed. He was right. It really has transformed things. I'd also like to thank Carl. He's incredibly responsive. If I have a technical issue with the software, he always gets back to me very quickly. So thanks, Carl, and thanks everyone for listening.